Hello, we're going to talk about microorganisms and how they can grow in different environments. What factors will affect their growth in different environments? So this is just a picture of different colonies grown on a petri dish, and as you can see how beautiful those colonies look like on a nutrient agar plate and just before we start. So to talk about the factors that affect microorganisms in the soil system, we need to keep in mind that microorganisms are living creatures. They are affected by various factors that affect any living creature. Mainly, if we're going to mention them, the type and the amount of nutrient is a major factor. Availability of moisture, and moisture is crucial for every living being. And then the degree of aeration, how much air is available. Microorganisms, they range from their oxygen preference and tolerance from aerobes to anaerobes. Then the degree of aeration and the temperature. Temperature is a very important factor that affects the survival of certain organisms, certain species in one location. The pH is also an important factor. And then when we talk about the soil, any factor, any management strategy would also affect what microorganisms grow in it, like applying manure or even enduring flooding systems. If we talk about aeration, there's a correlation between air and water. What you see here, what you see up here is these are soil aggregates, and within the soil aggregates we have the pore spaces, the white ones and the blue ones here. Those pore spaces, they are either filled with water, that's the blue here, or filled with air. And there's a correlation between how much space, how much oxygen is available for aerobic microorganisms. So oxygen concentration and their fluxes in the soil system is also affected. So in this area here, and if we can monitor how much oxygen is available here in the air system, just before the line where the water will start to fill the pore spaces, we can see a drop in the oxygen level concentration that will be available for cells. So as the pore spaces in the soil become filled with water, there will be less air available for aerobic respiration. And that is another demonstration here in this table, where in one of the experiments here done by certain professors, that concentration of oxygen and carbon dioxide, how they are different in tropical soil under wet and dry conditions. So what they did here is that they went through different depths from the surface. This is 10 centimeters from the surface, 25 centimeters down from the surface, 45, 90, and 120 centimeters below the surface level of the soil. They measured the oxygen percentage, the oxygen content, and the carbon dioxide concentration during wet and dry conditions. Wet and dry conditions. So what we can see here is that the numbers of oxygen concentration during dry conditions is getting closer to what we notice in the atmosphere. 21 is the percentage of oxygen concentration in the air, and within the 10 centimeters it's just similar to what we find in the air. It's around 20.7. As we go deeper during the dry season, in a dry soil, we can see that there's a decrease in the oxygen concentration. That is also followed by the same trend we see when we see a wet condition. But the difference is that during wet conditions, the available oxygen concentration, or the available oxygen content here during wet condition, is less than it is during the dry conditions. Now let's look at the carbon dioxide. The carbon dioxide percentage during in the atmosphere, in regular atmosphere, is about 0.03%, right? And during dry conditions, this is also the topsoil is a little bit higher, okay? But we could see also there's a trend of increase of carbon dioxide condition as we go deeper in soil. There's an increase in CO2 content. This is also evident in the CO2 concentration even during wet conditions, and they're much higher because of the microbial activity in the subsoil, but due to the consumption of oxygen and the release of CO2 that is merely trapped within the soil environment. So if we talk more about the effect of oxygen, prokaryotes display a wide range of responses to molecular oxygen. So what we mean by that is that we have different environments on Earth, and on which there are different levels of CO2 concentration, ranging from merely the similar concentration to what it is on the air, or to perhaps zero levels of oxygen concentration. But in between, there's also a vast array of different concentration, a gradient of oxygen concentration. And we could see different microbes occupying similar or those places. So according to the oxygen preference, microbes are grouped into obligate aerobes, which require oxygen for growth, obligate anaerobe, they don't grow with oxygen, it's obligatory. And we can see the facultative anaerobes. Those groups of organisms, they have the ability to switch between aerobic and anaerobic metabolism. And the aerotolerant anaerobes, they are insensitive to oxygen. We have another group where they're microaerophiles. They require less concentration of oxygen for their growth. So we talked about those obligate aerobes. They grow on the top of the test tube, closer to the surface, so they can be closer to the air. While we see the second one here, this is the facultative anaerobe, that can switch between aerobic and anaerobic type of metabolism. And then the aerotolerant as well. It has either also the capacity to tolerate oxygen. The oxygen is not toxic. And then we go to the fourth test tube, and you can see that those cells here, they are growing at the bottom of the test tube, escaping, getting away from any level of oxygen, even the ones that are dissolved in water. These are the strict anaerobes. And then the microaerophiles that we talked about, they need smaller quantities of air, okay? They grow not on the surface, but a little bit down here, where they can have enough oxygen that is suitable for their habitat. Now the question arises is that what makes those different species differ in their ability to tolerate oxygen? The answer is in their genetic makeup, in their ability to secrete enzymes or to produce enzymes that could help them regulate the free radicals originating from the aerobic respiration process. We mainly going to show you the effect of superoxide dismutase, SOD here, and the catalase enzyme. If the cells have the genes to produce them, if they are positive for superoxide dismutase and the catalase enzyme, then this cell is capable of controlling the harmful effect of free radicals forming during aerobic respiration. Facultative anaerobe as well has the capacity to switch between aerobic and anaerobic. The aerotolerant, they have the genes for superoxide dismutase, but not the catalase one. So it's positive for SOD and it's negative for catalase. The anaerobes, on the other hand, they don't have the genes for these two enzymes. Therefore, they're not able to tolerate the toxic effect of oxygen. The microaerophiles also have the SOD but have low levels of catalase enzyme. As we proceed to the effect of soil temperature, here we refer to a very crucial factor for growth of many microorganisms, where it could shape their ability to live in a certain environment. Even though microorganisms have been found growing in virtually all environments, any environment where there is liquid water, we will be able to find microorganisms regardless of their temperature. But that applies to the different varieties of microorganisms, different varieties of bacteria, who have different abilities to withstand temperatures. Microorganisms are found growing in sub-zero soils, in the Antarctic soil, to as high as 115 degrees Celsius in deep sea hydrothermal vents. So they vary greatly in their ability to withstand temperature. This curve here represents the temperature variation versus the growth rate. Okay, so here's the temperature versus growth rate. As we notice here is that the minimum temperature where microorganisms starts to grow, okay, they start to ascend, their growth rates start to become higher and increase until they reach what is called the optimum temperature. Optimum means the most suitable for growth, and the growth rate is maximum for them, which would be this area here. This point will be the optimum temperature at this point, after which also we will note a decline in the growth of the microorganisms as the temperature. This is for a single species. So there's the minimum tolerant temperature and a maximum tolerance, after the maximum and below the minimum, there is no growth for this microorganism. What applies for temperature also applies for pH, and we'll see that in a minute. Therefore, according to their preference of the microorganisms, they can be grouped into what we call them the cold-loving microbes. They're called saprophiles. They grow in a range of 10 to 20 degrees Celsius with optimum at 10 to 15 degrees. The second group are the mesophiles. These are the medium temperature loving ranges between 20 and 40. Optimum is 37. And note that most of the pathogenic human pathogens, they are mesophilic. They prefer temperatures as it is at our temperature, our body temperatures. And then we can note the thermophiles. These are the high temperature loving. They like high temperatures between 45 and 70 degrees Celsius. Their optimum is 60 degrees. And the extreme thermophiles, we recently came to know that some microbes tend to like they like to grow at very high temperatures between 80 and 120. So they are very much different in their temperature range. In this diagram here, we note the temperature. Okay, so this is the temperature range, also the growth rate, and you can see that we can group them into saprophiles, psychotrophs, a little bit mild cold loving, mesophiles, thermophiles, and the hyperthermophiles or the extreme thermophiles. They can grow at temperature closer to boiling temperatures. And this is unique because they could adapt a mechanism where their proteins could stay intact even at temperatures that high. Moisture is, we said, it's crucial for life. There is water, there is life. No water, no life. So the water availability is very crucial factor that affects the life of many cells. Mostly at different water activity levels, we can find microorganisms living somehow between 1 and 0.7. Agricultural soils range usually between 0.9 to 1. Those microorganisms who like to live in a very extreme dry conditions, they are known as xerophiles. Okay, so they can tolerate really low level of water activities. The soil pH is the hydrogen ion concentration. We all know this. It's natural environments vary. In nature, we can find environments where the pH level is 0.5, very acidic soils, to about 10.5 in the most alkaline lakes. Most free-living prokaryotes, they can go over a wide range of also pH. According to the preference of pH, microbes are classified into acidophiles, neutrophiles, and alkaliphiles. This is the typical pH curve, where at 7 is the neutral, here the neutral pH at around 7. Below this value is the acidic environments, and beyond this value is the alkaline environments. So those who prefer neutral pH, we call them neutrophiles. Those who prefer acidic conditions, we call them acidophiles, and those who prefer alkaline conditions, we name them alkaliphiles. So the acidophiles, the optimum pH well below neutrality. We have examples like the thiobacillus and the sulfolopus, thermoplasma. The thermoplasma, what's unique about it is that it's obligate acidophilic. It cannot grow in neutral conditions. They are obligate acidophiles. Neutrophiles, such as Bacillus and the Pseudomonas and so many genera as well, they grow best in neutral pH, and alkaliphiles usually prefer alkaline conditions. Here's an illustration where, again, growth rate and the pH level on the pH scale. We note here the minima and the maxima, point, minima and the maxima here, and here is the, for example, for this acidophile, this is the optimum pH. For neutrophile, optimum pH lies in here, and for alkaliphiles, the optimum pH lies somewhere here, and that's how they're classified. For the salinity, the response to salt, also microorganisms are classified into halophiles that require some sodium chloride for growth. Mild halophile require 1 to 6% NaCl. The moderate halophile 6 to 15% sodium chloride, and the extreme halophiles are found in extremely salty lakes and environments between 15 and 30% NaCl. And one example could be the Dead Sea, for example. It has bacteria that are known to be extreme halophiles. Organisms live in environments high in sugar. It's called osmophiles. So they could have special mechanism to regulate their activity in high sugar content. For metabolic classification, depending on carbon, energy, and the electron donors. As for the carbon source, we have two main mechanisms, whether they can use organic compounds, like we name them heterotrophs, they rely on readily available organic material, or on the other hand, they can synthesize their organic material from inorganic sources, such as the autotrophs. So the heterotrophs, they rely on readily available organic material, while the autotrophs depend, or they can synthesize their own from inorganic carbon. The energy source could be from the sunlight, we call them phototrophs, or it can use from chemical bonds and chemical reactions, we call them chemotrophs. Electron donors could have inorganic sources, inorganic compounds could become an electron donor, so we call them lithotrophs, or if they come from organic compounds, we name them organotrophs. And this is an introduction to different metabolism and metabolic classification. So, for example, the lithotrophs here, the lithotroph or the chemoautotroph, they use inorganic energy sources like ammonium ion, nitrite, sulfur ion, okay, and carbon sources like CO2, as also inorganic sources for electron donors. So they're called chemoautotrophic. Now the chemoheterotrophic, they use organics for energy, organics for carbon, and organics for electron donors. With the photoautotrophs, of course, photo means they depend for energy on light. But when it comes to the carbon source, the autotrophs, they use CO2 in their as the primary carbon source, and the photoheterotrophs, they use organic compounds. Okay, and photoheterotrophs, sometimes they also use organic compounds as electron donors. Thank you.